And, I'll be amiss not to thank Grażyna and Anna and Tim for putting all of the effort to get this seminar successful. So thank you very much. There's not enough thanks to really give it to three of you, but I will try my best. So thank you very much. Again, we have another wonderful science discussion today on lipids, and I will just move to Jasmine. Jasmine.
Great.
Good morning, Chairman. We were just calling your name, but.
Yes, I was finishing up another meeting, but glad to see you all and looking forward to the best hour of the week.
Alrighty. Good morning, everyone. Hello. Welcome to today's seminar series. Today, I have the pleasure of introducing our guest speaker, Dr. Julia Busik. Dr. Julia Busik obtained her PhD at Graduate University for Advanced Studies in the Department of Physiology in Yokohama, Japan. Soon after, she became a visiting graduate research at Michigan State University in the Department of Physiology, where she developed into her professor with tenure position, which she holds today. Today, she is speaking from the Department of Physiology from Michigan State University. Dr. Busik currently is the chair of the College of Medicine Reappointment, Promotion, and Tenure Committee, and an active member of the Molecular Metabolism and Disease Research Group. She has served as an associate editorial board member for various journals and as a manuscript reviewer. Dr. Busik has also done consultantship for companies such as Takeda. From her first award of gold medal for excellent study in high school back in Russia to her most recent award as Silver Arvo Fellow for her contribution to the Association for Research in Vision and Ophthalmology, Dr. Busik's cutting-edge research has resulted in over 90 publications. Her lab has facilitated the training and development of countless high school, undergraduate, graduate researchers, and postdoctoral fellows. Please help me in welcoming Dr. Busik.
Thank you so much for this introduction. And can I share my screen and go ahead?
Please.
Okay. Do you see my presentation screen?
Yes.
Yes.
Perfect.
Okay.
We're seeing your disclosure slide, if that's the first one.
Okay. Yeah, that's the first one.
Okay.
My presentation today will be lipids, retina, and diabetes, the good, the bad, and the ugly. And I promise I will show all the different lipids in there. And I will start with the disclosure. I am CSO of Ceramides Inc. But I'm not going to speak about the work that I do for Ceramides today. So first of all, I will introduce diabetic retinopathy. Most of you probably know that it's disease of the back of the eye, where in healthy retina, we have well-organized vessels with metabolic abnormalities. We get changes in the diabetic retina that lead to blood vessels that are leaky, and that could eventually lead to blindness. It's a recent study conducted by CDC in 2020 found that prevalence of diabetic retinopathy is still very high, that in adults over age of 40, we have 1/3 of Americans, especially in African American and Mexican Americans with diabetes who have retinopathy. So it's 30% of retinopathy patients over age of 40 do have some degree of retinopathy, and that accounts to about 4.2 million adults with retinopathy and 655,000 with vision-threatening retinopathy that could lead to blindness. So it's clearly a very important disease to understand the pathogenesis and to find treatments, with the numbers still increasing for diabetes and retinopathy. And I would like to first give you a bit of a historic perspective of what do we have already and what do we know about it. So the treatments for retinopathy started with the first clinical trial in 1968. It was Early House Symposium, which is here. Late Lloyd M. Ayala is presenting at the last meeting, talking about that first clinical trial, which was a diabetic retinopathy study when the laser photocoagulation was used, and at 4 months intervals and after photocoagulation, the vision improved and the loss of vision was prevented for more than 5 years in the patients. And that was really quite revolutionary at that time, and we did see that became a mainstream for treatment of retinopathy. However, this slide is actually, I stole it from Lloyd P. Ayala, who was presenting at ARVO last year as an award presenter, and he did mention that his father was always saying that, how come that we consider this one a success and this one a failure? That we can do better. That's really, we are preventing some of the damage, but we can still do better for diabetic retinopathy. So what did we do since that time to do better? First of all was DCCC trial, the Diabetes Control and Complication trial, which showed that tight control of blood glucose significantly decreases risk of diabetic retinopathy. These are just the two main outcomes for conventional versus intense control of blood glucose in patients who didn't have retinopathy at the beginning of the trial, or who had some degree of retinopathy at the beginning of the trial. You can see that there is a slight increase that we often see in diabetes when the control is reestablished, but then there is a conventional group continues to increase, but intensive group actually declines after. And the rates of prevention are very similar in patients who didn't have or had retinopathy to start with. So from that time on, we knew that tight blood glucose control is important, and that's not questioned, and that became a saving vision treatment for many patients to just have a better control. And further basic science translating to clinical success was the intravitreal anti-VEGF therapies, as you all aware of, that started in 2004 with Aptamer, and then the monoclonal antibody in 2005, and Fragment in 2006, and the fusion protein in 2011, and it continues to develop. And all of these agents, we know that they prevent leaking blood vessels, they prevent edema, and they slow down development of neovascular retinopathy. However, we all know also that there are problems with VEGF, anti-VEGF treatments. About 40% of patients do not respond or don't respond as expected to anti-VEGF therapy. And it's directed at a very late stage of the disease when we are not expecting reversal, we are just preventing further damage. And it could be potentially detrimental at the long term. We start seeing some effects that inhibiting VEGF for a long time could have some effects on other tissues like RPE and the neuronal tissue in the retina. So great treatment, we have really improved from where we were before, but we can do better. We can do more things to further improve the treatments. So I just put in several lipid statements in here that we already looked at carbohydrate, which is the glucose itself. We look at proteins, activation with cytokines, activation with growth factors. However, lipid represents 25 to 30% of dry weight of the mammalian retina, and we didn't talk about lipids, and we didn't really look at the lipids as a modality for treatment of the retinopathy. Lipids present in the retina are highly unique, and they play a critical role in retinal function and disease. That's well known. And if we look at the clinical trials, there is a strong association between severity of retinopathy in type 1 diabetes and the size of the particles of serum lipoproteins, as well as LDL concentration. That's the work of Timothy Lyons, analyzing the same DCCT trial patient samples and showing that they didn't just have improved hemoglobin A1C, their lipids also improved. And well-known ACCORD-I study showed that combination of fenofibrate with simvastatin significantly slowed progression of retinopathy over 4 years when further glycemic control was not effective. However, it's not that simple. The blood glucose is relatively simple measure that relates to retinopathy control. Blood lipids do not quite do that. So the WESDR study showed no association between total cholesterol or high density lipoproteins and incidence of diabetic retinopathy. The beneficial effects of fenofibrate did not correlate with changes in serum lipids in field and ACCORD-I study. And the JAMA Ophthalmology and Diabetes Care studies that published this data both stated that tissue-specific retinal lipid regulation might be more important than systemic dyslipidemia. So with that, the drugs that we have now to control lipid metabolism, they are mainly directed at blood levels. However, they could control retinal specific levels. They also can have lipid independent effects. So today I'm going to focus on the retinal specific effects for the purpose of today's talk. We are not going to talk about the blood and lipid independent effects, not because they're not important, just focusing on what I can cover in the time that I have. So we will start with ceramides. You probably have all seen the Scott Summers paper, Could Ceramides Become the New Cholesterol? There are measurements of ceramides that Mayo Clinic is now adopting that seem to have better correlation with cardiovascular health than cholesterol levels. And that's also echoed in Samar Hamad's paper with plasma sphingolipids profile that associated with atherosclerosis and lupus erythematosus. So both of this, and there are more studies appearing, start to paint a picture that ceramides might be the next cholesterol where we could use it for both prediction of the disease, but also to control the disease. So when I talk ceramides, what are they? It's a sphingolipid. It's a sphingolipid that comes from sphingomyelin, which is the molecule of sphingomyelin it's shown here. It has a sphingosine bond, fatty acid attached to it, and phosphocholine as a head group. And when sphingomyelinases can cut off the phosphocholine group from sphingomyelin, we get ceramide molecule. We have shown before, and several laboratories confirmed that acid sphingomyelinase is highly upregulated in diabetic animals. Here we are using a ischemia reperfusion model of diabetic retina. However, we showed that in the diabetes model as well, in STZ diabetes, as well as type 2 DBDB mice. So acid sphingomyelinase activation is shown here. And if we use acid sphingomyelinase knockout mice, of course, there is no acid sphingomyelinase, nothing to activate. But what we see that all the inflammatory parameters that normally come up in diabetic retina, again, ischemia reperfusion in this case, the ICAM1, BCAM1, they are not increased in acid sphingomyelinase knockout animals. Alpha beta VEGF follows the same pattern. Again, they're increased in the wild type and they are not increased in the knockout animals. That's a publication that shows that. And also, if you look at a cellular capillary formation in the animals that are wild type for acid sphingomyelinase, we have an increase, a typical increase in a cellular capillaries. And we don't have that in acid sphingomyelinase knockout. That's Madalena Brana who did that study a while back. However, using the acid sphingomyelinase knockout here, or any kind of inhibition of acid sphingomyelinase would not be something that we would use in clinic. Acid-sphingomyelinase is a very important enzyme, and that's inhibiting acid-sphingomyelinase is the last thing that we would want to do in clinic. It causes Niemann-Pick-like disease, and it causes also neurodegeneration. So we wouldn't suggest the inhibition of that. However, we did use we could use the retinal vascular pathology when we inhibit acid-sphingomyelinase inside the bone marrow. So that's when we don't inhibit all of the neuronal acid-sphingomyelinase. We just do the bone marrow acid-sphingomyelinase knockout that is transplanted into the lethally irradiated animals. So all the bone marrow cells become acid-sphingomyelinase knockout, but the rest of it, the retinal cells, the neuronal cells are not. So in this case, we can have very similar to what I showed you before with the ischemic reperfusion in diabetic animal with the inhibited acid-sphingomyelinase in the bone marrow. We can have decrease in permeability, that's the first slide here, and decrease in the number of acellular capillaries. Another way of doing that, and this one I will disclose that this is the work that I'm doing with Ceramides now, but the study, when I did it, that was still I was not involved with Ceramides yet. So we used the anticeramide fragment that inhibits the formation of ceramide domains on the surface of the cells without inhibiting sphingomyelinase. So it inhibits that process that I showed you at the beginning, conversion of the of sphingomyelin into ceramide when the ceramide is formed and forms the platforms on the surface of the cell. Anticeramide antibody would bind to that and stop the propagation of the signal, but without inhibiting sphingomyelinase inside the cell. So we still can preserve neuronal health, the intracellular health, but inhibit the inflammatory signaling. And with that, this is a ischemic reperfusion slide where when we apply this anticeramide treatment inside the intravitreally, we inhibit the permeability, that ischemic reperfusion induces permeability after 2 days of post the treatment. With the antibody, we don't have that. And with that, most of the inflammatory cytokines that are upregulated are now normalized. TNF, IL-1 beta, IL-6, ICAM, VCAM, MCP1 all go down with the treatment with the anticeramide fragment. In diabetic rats, we had very similar picture where we treat them with anticeramide fragment intravitreally. In diabetes, we have an increase in leakage, and with the fragment, we now have downregulation of that leakage in the animals. I talked about the ceramide inhibition in the bone marrow cells and trying to inhibit ceramide in the platforms, but there is also another way to look at the ceramide changes in the retina. We actually do not see an increase in ceramide overall in the whole retina. However, if we isolate mitochondria from the endothelial cells in the retina, these are from the animals that were diabetic for 36 weeks, we see an increase in the shorter chain ceramide 16O and 18O in the mitochondrial fraction of the diabetic retinas. So is it acid sphingomyelinase, is it present in mitochondria? Do we really have it in there? And why do we see that? First of all, we had to show that we're isolating mitochondria, not lysosomes, because lysosomes have very high level of mitochondria and sometimes they co-precipitate. So we showed here then that we do have acid sphingomyelinase tracking with the mitochondrial fragments. And if we purify it further, when we dropping down the mitochondria, the lysosomal fragments, acid sphingomyelinase still stays in the mitochondrial fraction. So showing that, yes, it is expressed in the mitochondria, and that increase in ceramide that we've seen before could be from acid sphingomyelinase actually in mitochondria. And then to see if the ceramide in mitochondria could have an effect on respiration and could affect the cells, actually had to design our own microfluidic respirometry chamber, which was done in collaboration with Denis Proshlikov, where we could have adherent cells under the flow conditions and measure continuously their respiration to be able to see the effects of ceramide and sphingomyelinase treatment on the respiration in the cells. So this is our typical respiration trace, where we measure basal respiration and uncoupled and inhibited respiration in the cell. And at rest with the basal respiration, we have control or acid sphingomyelinase inhibited mitochondria have very similar rates. However, when we go to uncoupled respiration or more active respiration, acid sphingomyelinase inhibitor actually improves that active rate. And going back to inhibited, we have very similar rates between them. So the acid sphingomyelinase inhibitor or changes in ceramide in mitochondria does improve our maximal respiration rates, and that's showed here also in a graph form. Why is it important? Because when we treat the cells with cytokines or LPS in this case, we do have a decrease in basal rate, and that decrease in basal rate that will transpire into a decrease in the maximal respiration as well. So with acid sphingomyelinase inhibitor, we don't have that decrease, and we also have that increase in the residual capacity of the mitochondria. So in other words, when we have acid sphingomyelinase inhibition, we can restore the respiration of endothelial cells back to normal after we treat with cytokines, or if you look at it as diabetic mitochondria in the case of when we isolate them from diabetic cells. So there are several ways again to affect ceramide, and we could have it on the cell surface where we could have acid sphingomyelinase, anticeramide antibody, or we could have specific mitochondrial treatments where we could have acid sphingomyelinase in the mitochondria inhibited. However, all of that I was telling you about was about the bad ceramide, the 16O and 18O ceramide, which are very pro-inflammatory. They form platforms on the cells. They inhibit respiration in mitochondria, but that's not the whole story about ceramides. When we look at the vitreous of the patients with proliferative diabetic retinopathy versus non-diabetic patients, this controls had macular hole and the vitreous was removed for that. We see an increase in 16O ceramide, 24 and 21, which are all this pro-inflammatory ceramide species, at the expense of a big decrease in 26O ceramide, which is a very long chain ceramide. And when we first got this data, we didn't really pay attention to that, but then as the study developed, we noticed several other things about this very long chain ceramides that kind of piqued our interest. First of all, the short chain N24-1 ceramides, the ones that go up, they are all substrates for sphingomyelinase. However, sphingomyelinase activity drops almost to zero when you get to the longer chain. So the biology of this species is quite different. And the longer chain, they require very long chain fatty acid to be attached to them, to be synthesized. That could only happen with fatty acid elongases, making this very long chain fatty acids. And when we did this study, it was a while back, to our surprise, we noticed that retina has really high levels of elongases. That's compared to the liver. We have all the main elongases, level 2, 6, and 4, were very highly expressed. And liver doesn't express 4, and the rest of them were actually lower in the liver than in the retina. And what was interesting that in diabetic retina, all the elongases were downregulated. So we had almost 50% decrease in the elongase activity in the diabetic retina. So Maria and Todd did the study. So why is that important? The ILAVL4 especially is the enzyme that makes very long chain fatty acid. It can make monounsaturated or unsaturated fatty acids through de novo lipogenesis. That's our 26O and 28O. It can also make polyunsaturated very long chain fatty acids. I'm not saying that the polyunsaturated are not important. I'm just not focusing on them today. This is actually also a very interesting study, and there is a lot going on very long chain polyunsaturated fatty acids, but for the purpose of today's talk, we will focus on this very long chain ceramides and saturated fatty acids. So the very long chain saturated fatty acids will be mainly incorporated into ceramides by the action of ceramide synthases, and ceramide synthase 2 and potentially 3 are the ones that will be doing this job. So when we have ILAVL4 on board and we have this very long chain fatty acid, it can be attached to sphingosine bond by ceramide synthase, making very long chain ceramide. When we don't have it or it's downregulated, we go back to this shorter chain ceramides that are more inflammatory. So that's a kind of to summarize what I just said about this different molecules. With ILAVL4, we have very long chain ceramides. In diabetes, we have less ILAVL4, higher levels of sphingomyelinase. We have this shorter chain ceramide 16O, 18O. The longer chain ceramides could be further acted upon to make omega hydroxy ceramide, making this bond, and that's this kind of a very strange looking structure, which we didn't know whether it was present in the retina and what it's doing, but I'll be talking about it a bit more. When we overexpress ILAVL4, we have this kind of change in ceramide species. This is in endothelial cells. We have downregulation of 16, 18, and 24-1, and upregulation of 26O ceramide. If you remember what I showed you about the vitreous samples from non-diabetic versus PDR, we can see that this is a mirror image. So when we have higher levels of ILAVL4, the ceramides that go down in diabetes actually come up, and the vice versa, the ones that are going up in diabetes are coming down. So what are these very long chain ceramides? And again, why are they in the retina? What are they doing in the retina? And we previously, this very long chains omega hydroxyceramides were found in the skin. They provide the water barrier and prevent dehydration. And actually most of the ILAVL4 knockout animals die from dehydration, or if we have ILAVL4 related macular phenotype, the patients would also have the dehydration of the skin. It's a Stargardt-like macular degeneration that's caused by ILAVL4, and again, skin is one of the problems there. So the question was, is it also preventing permeability, decrease in permeability, and does it provide barrier in the retina as well? So we hypothesized that endothelial cells in the tight junctions could have 3-omega hydroxyceramides stabilizing tight junctions. To do this work, we started collaboration with David Antonetti lab, and that was Norman Cady was the one doing this study, and a lot of it was done in David's lab actually. So what we did, we looked at the overexpression of ILAVL4 when we have cells, these are bovine retinal endothelial cells. When we treat them with VEGF, there's a disruption of tight junction barrier. So this is ZO1 expression in tight junctions. You can see the nuclei in blue and tight junctions are in magenta here. And when we have cells treated with VEGF, there is disruption of that barrier. You can see that it's very jagged. ELAVL4 by itself didn't do anything to the barrier, but when we overexpressed ELAVL4 and treated with VEGF, the barrier was normal. We did not have the disruption. We also looked at the ceramide expression with anticeramide antibody, and we see ceramide also expressed in the barrier, actually overlaying perfectly with the ZO1. So that gave us a thought that ceramide might be in the tight junctions. Of course, there is ceramide intracellular as well, and the way it should be, that's a lysosomal ceramide and in other structures, but we are looking now at that barrier one. And when the barrier is disrupted, ceramide really is moving out of the membranes, and you can tell that it's disappearing from that structure. And when it's treated with ELAVL4, we do see, again, normalized barrier, normalized ceramide expression in these cells. To see if ceramide is really in the barriers, do we have it in the tight junctions themselves? We first did electron microscopy. This is endothelial cell tight junction. You can see that's a higher and higher magnification. So that's a tight junction in the retina, and it's a larger magnification of this one, and then going further, and that's a kissing point in there. To do the electron microscopy with the gold particle study, the immunoelectron microscopy, of course, we don't get as beautiful pictures, but we would usually find a slide where we would look first, find the junctions, and then look at on the next one, we would do the immunogold staining. And here in immunogold, that's a tight junction. And we are staining it with occludin to see the larger gold particles and ceramide in the smaller particles. And you could tell that there is a ceramide right in the junction. So that gave us hope that it's probably expressed in the tight junctions inside the junction, as we are predicting that it could be stabilized in the junction. You see it, of course, inside the cells as well, because as we should. Then to really prove that we do have that in the junctions, and that could be, the hypothesis could be correct, we first actually isolated tight junctions, and we used mouse dermis as our control, because these structures are not available as a standards, as the internal standards. So we isolated mouse dermis to make our own internal standards. And this 264-2682 peak is the one that corresponds to the structure. And we do see it in tight junctions. They are here isolated from AAP19s because we needed a lot of cells to get the tight junction prep like this. But yes, we did get it out, and it's in there. This is just the solvent peak. Don't pay attention to that, but this peak is the kind of the peak that we are looking for. So that showed us that yes, we do have this ceramide and tight junctions, and that could stabilize it. When we went into the animal study and did very similar delivery of ELAVL4 into the diabetic animals, and as Dave and Taneti's lab showed many times, the tight junctions are disrupted in the diabetic animals. So this is control with ELAVL4 overexpression, control with empty virus, where we're looking at occludin expression in the vessels. And when we overexpress ELAVL4, we have the normalized looking occludin expression with tight junctions that are not as jagged and disrupted as in diabetic animal. And with that also, the overexpression of ELAVL4, where it was improving the permeability increase that we see in these animals. Again, control, normal permeability, control with ELAVL4, no changes, high permeability increase in diabetic animal. because of the disruption and normalized in level for animals, and that's a quantification of that. So that's the good ceramide. And now, just to summarize this, so if we can have E level 4 attaching the ceramide and making 3-omega hydroxy, then we could have normal tight junctions, which in diabetes are replaced by the long chain, by the short chain ceramides, leading to barrier breakdown and leakage. And with that we can have increase in permeability, and to improve that increase in permeability, we could have E level 4 or increase in VLC ceramides, or we can inhibit sphingomyelins and short chain ceramides, and either one of those could lead to could improve the outcomes in retinopathy. Now we will switch to cholesterol. That's my ugly one. So cholesterol uptake and endogenous cholesterol biosynthesis are very essential for normal retinal function. Retina needs cholesterol. Cholesterol is needed for the photoreceptor function, for visual cycle, and that's why cholesterol is constantly recycled, and that recycling is as controlled as the liver whole body recycling of cholesterol, and probably even more important for the retina. In diabetes, what happens is that we have, first of all, the downregulation of metabolism of cholesterol to oxysterol. So we are removing that oxysterol removal of cholesterol from the retina, and with that we are removing LXR. I went too fast. We are removing that LXR regulated ABCA1, ABCG1 upregulation and removal of non-oxidized cholesterol from the retina. In addition, we have the blood retinal barrier breakdown and increased permeability of vessels to cholesterol, which will also lead to cholesterol accumulation in the retina. With all of that, we have higher levels of cholesterol in the retina, and that's just shown in one of our studies when we have non-esterified cholesterol increase, which can eventually lead to problems. And one of these problems, which I'll try to convince you, is formation of cholesterol crystals. Cholesterol crystals in disease in the retina have been studied for a while, since the spectral domain OCT started showing structures that look like crystals. They're called hyperreflective crystalline deposits. These are from AMD, and this is from this paper by Christian Kirschner. There are more papers in different diseases to show that crystalline deposits can be a predictor for both macular degeneration and as well as diabetic retinopathy. I'll show it a bit later. And there's this CAM report 5, which describes these features and how they form, and suggests that using crystalline deposits as one of the predictive values for disease progression. It's also highly upregulated in Coats disease, and this is the paper that shows that in diabetic macular edema, the hyperreflective structures could be predictive of response to drug and progression of the disease. So this is by OCT, and it's been developing over the last several years to predict what structures they are and how to analyze them. However, we cannot tell for sure what they are from OCT. We don't know if this is cholesterol, or we cannot analyze it from OCT structures. Because of that, we started looking in the animals by SEM, scanning electron microscopy, with the analysis of the structures that we can do inside the scanning electron microscope. So this is my background retina is not just for fun. This is actually the actual retina that we took, the picture of SEM that we took for our animals. This is a diabetic animal, and done, first of all, the one that I'm showing in the background is done with all of the steps required for electron microscopy. However, if you do that, you dissolve lipid. If you do critical point drying, or if you do any kind of ethanol treatment, you dissolve lipid and you're not going to see any of the lipid droplets. You're not going to see the cholesterol in there. If we don't do these steps, the retina doesn't look as pretty. You lose the fine structures, but you start seeing what we call a lipid pools. Like you can see them pulling up here. And I lined it up so that it kind of corresponds to the structures where it would be on the well-preserved retina. And inside the structures, if we enlarge them, and I colored it here for you so that you could see it better, these lipid pools that are now in olive color are full of this glass shard-like structures, which are cholesterol crystals. And we know that these are crystals because, again, in the electron microscopy, we can analyze inside the machine, we can analyze the structure and see what it's made of. And then there are only several things in the retina that can crystallize. It could be monosodium urate, it could be calcium crystals, could be salt or cholesterol crystals. And by composition, this is all carbon and hydrogen, so it's really not any other crystal could be in there. And they are highly upregulated in diabetic retina. We really don't see them in the control animals. We have also done analysis now of human retinas from cadaver eyes, and we do see these crystals in the retinas from the type 2 diabetic patients. We didn't do any type 1 yet. Not patients, sorry, the cadaver eyes. So what's the problem with it? Why this crystal formation? Why am I talking about it? And why am I calling it ugly? Because cholesterol crystals can induce a lot of tissue damage. They are taken up by the cells and they are put on the lysosome by the process that's called frustrated phagocytosis, because the cell is recognizing it as foreign body and tries to digest it, but cannot. And that activates the proteases, complemosome, NLRP3, inflammasome, and cell death, as well as inflammation. So all of this can happen in the diabetic retina from these cholesterol crystals. To show that we can form crystals, if you leave cholesterol to dry, it will form crystals. And if you have water inside it, it would be this monohydrate crystals, which look like this glass plates, or pure crystals without water that look like needles. And either one of these will lead to inflammatory changes. These are in retinal pigment epithelial cells. These are human IP, not AIP19s, but like regular human IP cells. And IL-1 beta 8, MCP1, IKM, PKM, and the complement 5A receptor were all activated by treating the cells with crystals. And they can also increase cell death and inflammation in retinal endothelial cells. This is bovine retinal endothelial cells. And we see very similar picture with cytokines. And we see that caspase-1 is upregulated and cell death is upregulated by the cholesterol crystal treatment of the cells. Interestingly, statins, alpha cyclodextrin, or fibrates, phenofibrate, can dissolve crystals ex vivo. If you treat crystals with any of these drugs, which is cyclodextrin, atorvastatin, rosuvastatin, and phenofibrate, they all dissolve crystals. Hope you can see it, like this is before you apply a number of crystals, and that's the same kind of place after you treat with the drugs. And when you treat with all of these same compounds in the presence of crystals, this is shown from endothelial cells, but we've done it also in IPE cells, that we have decrease in inflammatory as well as complement activation. I'm just showing IL-6 here for simplicity, but we did all the panel of cytokines and they all show similar picture. And what was interesting that phenofibrate actually does better job than the statins, which could indicate that potentially, again, not quite claiming it yet, but potentially that phenofibrate effect in the retina was through dissolving of the crystals, because it was almost as good and sometimes better actually than the alpha cyclodextrin, which is known to dissolve crystals. We then went and did the in vivo study using alpha cyclodextrin, because that's one of the molecules that's being used in clinical trials. These are all clinical trials with alpha cyclodextrin. It's quite safe and it does have the cholesterol dissolving power. So we treated animals with alpha cyclodextrin and showed that it does dissolve cholesterol crystals in the diabetic retina. So first of all, this is just cholesterol levels. This is unesterified and esterified cholesterol, where we have normal levels. We do, as we talked about it, we do need cholesterol. So normal levels of cholesterol are here in the middle, and the diabetic animals have higher levels of cholesterol in pretty much all the layers. And you can tell that with alpha cyclodextrin, they all go down to the control levels, if not lower. And for esterified cholesterol, so we have in diabetic animals, we have increases mainly in the barriers, and it goes back to normal levels with alpha cyclodextrin. And what's important, that we had no cholesterol crystals in the diabetic animals that were treated with alpha cyclodextrin. And that could be done as a prevention. Even after 2 weeks of treatment with alpha cyclodextrin, we start seeing changes, and after 2 months of treatment of the animals that were diabetic for 6 months, we see disappearance of these cholesterol crystals in the retina. So to summarize this part, that diabetes leads to non-esterified and esterified cholesterol accumulation in the mouse retina. Cholesterol has propensity to crystallize, which can lead to crystal formation in the retina. And treatment of cholesterol crystals is pro-inflammatory and causes cell death in both inner and outer barrier cells in the retina. And alpha cyclodextrin dissolves crystals and prevents, both in vitro and in vivo, and prevents the inflammation and complement activation in the cells. And interestingly, it's not just alpha cyclodextrin, it's statins and fibrates, with phenofibrate really doing a good job dissolving the cholesterol crystals. So to conclude, all of what I was talking about today, I hope I convinced you that normalization of lipid metabolism could provide a new direction in the development of therapeutic strategy for prevention and treatment of diabetic retinopathy. And with that, I will thank my lab and people who did all of this work. So a lot of the studies I showed are done by very talented postdocs, Sandra Hammer, and graduate student team, Dot Weller, Norman Cady, Madalina Apriano, Maria Tikhanenko were doing part of the studies that I had in there. And Todd Leidig is my mass spectrometry guru, who is helping a lot with all the lipid analysis. George Abella is a cardiologist who introduced us to cholesterol crystals. The scanning electron microscopy, I also have to thank Maria Grant and Irina Pikulova for the, we're working together on cholesterol crystal work with them, and Dave Antonetti's lab for the studies on the permeability barriers. Rich Kolesnik and Zvi Fuchs from Memorial and Sloan Kettering are helping with Isis Singamalny's story of that. And Denis Proshlikov is the, we work on mitochondria with him. And I promised to show some snow and what we do with my lab when we are not in the lab. So that's what we have in there. And with that, thank you. And thank you to my pups for staying quiet and letting me present today.
Thank you very much. We have several questions, so let's start with Philip Kaiser.
Hi, Dr.
Professor
Busik.
Kaiser, are you there?
I am here. Thank you. Dr. Busik, great presentation. I really enjoyed it. Just
you.
two quick things. The first goes to the crystal study that the cholesterol crystal study that you performed. So I was kind of I was just surprised to see that those compounds like atorvastatin, drugs that are inhibiting, enzymes involved in cholesterol biosynthesis are so effective in dissolving those cholesterol crystals. And it was kind of, chemically diverse things could do that. So I was wondering if you had any insights into the mechanism of why those are effective. And then the second one, it goes back to the work you were showing with the anticeramide antibody, which I've been pretty interested in that approach. And I was wondering how effective, how much of the ceramide can you actually extract out with the antibody? Because it's a little surprising that it works well to me, given you have something that's sort of embedded in the membrane, and kind of how accessible it is and how much you actually get into solution that would be accessible to the antibody. Thank you.
Both great questions. So the first one with the cholesterol, it's the effects of the drugs. If you noticed, I'm only using the statins which are lipid soluble statins. There are water soluble statins which don't work that way. They don't have the same effects. And we have several studies already that they do not work through the same HMG-CoA mechanism. If you just put cholesterol and put the atorvastatin or rosuvastatin on top of it, the cholesterol crystals, it will start dissolving it. You just see the drug dissolving it. Same with alpha-cyclodextrin, same with fenofibrate, which I was really surprised by, that fenofibrate does the same job. And how does it do it? We actually have a chemist we're working with right now looking at that. So it's probably a relatively non-specific mechanism, because these are all lipid-soluble molecules, and they just bind to the crystal and help the crystal to kind of almost eat up from the surface of the crystal. That's what alpha-cyclodextrin does. It's just this conical shape with a lot of hydrogen bonds that just bind to it and start binding off the molecules. That's probably the same. That's all I can tell at this point. We don't know exactly why, but again, I was really surprised that fenofibrate was doing that. But when you look at the structure and at the fact that all of these molecules are lipid-soluble molecules, it kind of starting to make sense. They're hydrophobic, they're lipid-soluble. When they are in close proximity to the crystal, they probably have high affinity for crystal than to water around it, and they can start dissolving the crystal. Does it make sense? So that's as the best I can say at this point. But again, we're working with several chemistry professors to see what exactly could be going on there, and if we can maybe design something that can dissolve them better. So alpha-cyclodextrin is doing quite good job on that. So the second one with the anticeramide antibody. The ceramide itself, if it's normal levels of ceramide, will never be accessible to antibody. It's inside the membrane without the head group. It's not out enough to be accessible to the antibody. For ceramide to be accessible, you need to have the ceramide-rich platforms. And ceramide-rich platforms is when they form, ceramide has a shape of the cone. They start having that invagination shape, and we don't know yet exactly. That's a big question for the company also. What exactly does the antibody bind? We don't think it binds one ceramide molecule. It binds the shape. It probably binds the whole domain, because if you think about the size of the antibodies of one ceramide, stoichiometry will not quite work. So it's most likely binding several ceramides when they are forming the platform, which is actually a good thing, because ceramide is important. It's a structural molecule. We don't want to remove ceramide. We only want to remove the platforms that are forming when you have inflammatory signaling. And it's chronic inflammatory signaling that would lead to formation of these domains, because the fast activation of with cytokines that would happen with inflammatory, let's say, bacterial infection, it resolves very quickly, because these domains, they're very, they are temporal, they will form and they will resolve. But when you have chronic inflammation, like we have in diabetes, that's when you start having these domains persistent and being on the membrane for longer duration, and they could start causing damage. And that's what the antibody is targeting. We are doing several studies. We're actually looking at prior EM now, exactly to answer your question, to see what exactly do we bind? How many molecules of ceramide do we bind? But by functional data, we do know that we can, I have some data when we show the formation of these platforms, when we fix the cells after we treat with cytokines, and if we apply antibody, these platforms are not there anymore. So that's on the functional level, I can answer that. On the level molecule to molecule, invite me a year later, I will have an answer, hopefully.
It has a good point. I have a different point from a second question. Even if this thermodynamically is possible, did you have a calculation that whether affinity of the antibody toward whatever platform or individual lipid is sufficient to rip off the lipid from the membrane? As you know, lipid modification of protein brings protein to the membrane, and there's no way of extracting it unless you disturb that with, let's say, low ionic strength or any other ways. And that can be done. Here, you talk about platforms, so you have multiple lipids embedding in the membranes. So I just don't believe that there is enough power, thermodynamic, energy that is derived from the antibody binding. I think it's just an artifact of some sort.
Sorry, we are not trying to remove it from the membrane. That's exactly the point, that we are not trying to, thank you for bringing that up. We are not trying to remove it from the membrane. When the antibody binds, it changes the stoichiometry of the binding between the ceramides themselves when they are not forming that platform anymore, in a way that there's a sitting on the ceramide prevents ceramide from making these very tight platforms. That's what we are seeing. We are not trying to take it out and rip it out of the membrane, which I totally agree it wouldn't be possible.
Jim Howe.
Yes, thank you very much. That was a lot of fun. So the comment that I have is that it struck me that cholesterol has a high head group area to hydrocarbon volume ratio, whereas ceramides have a high hydrocarbon volume to head group area ratio. And I'm wondering if you're kind of getting a tune thickness of the membrane based on these ratios, and if that discrimination might be part of the problem. There's membrane thickness, just to take the simple idea. And the other thing that struck me was, I wonder if you've looked at your lipid effects in AMD. You touched on that a little bit.
I will start with the last question. No, we did not look at the effects in AMD. We are working with Kristen Kershaw, and she really wants to do that. So that's probably going to come, but this, as you can imagine, very time-consuming studies. And we just started the retinopathy. This is actually, we got the grant about less than a year ago. So we just started the studies, and we hopefully we'll get into that. In terms of the thickness, I want to make sure that I understand the question correctly. Are you thinking about the thickness of the double layer membrane, or the thickness or inside this little domains that are forming? Like
I'm thinking of the overall average thickness of the hydrocarbon region of the membrane, so the double layer.
So in the double layer, that will not affect the thickness, because both cholesterol and ceramide are spacers in a way. They will be in that phospholipid bilayer within the hydrocarbon chains. They will not go to the head groups. Both of ceramide and cholesterol, they get kind of stuck in between those, the double bonds, and not double bonds, I'm sorry, two fatty acids on the phospholipid. So they really don't stick out normally. Where your question is totally correct, where the thickness could be affected is in the cholesterol-rich or ceramide-rich domains. Normal lipid rafts, normal the caveoli will have both ceramide and cholesterol, and the interdigitation of ceramide and cholesterol will make stable membrane, which is very important for signaling and very important for normal dynamics of the membrane. And that one is thicker because of cholesterol is present in there. When you have ceramide-rich platforms, when it's only ceramide, it forms this almost like one layer invaginations in the membrane that could be so thin that they can lattice through. They make a hole in a way. So yes, there is this crosstalk, and high cholesterol, high ceramide levels in these domains was shown to push out cholesterol. So you could have that dynamics as well. So yes, like for the rafts, yes, for the phospholipid bilayer, that's probably wouldn't quite be.
Thanks.
But thank you. Yeah.
All right, Dorota.
Hi. Hi,
Hi.
Dorota Skowrońska-Krawczyk. I admire your work, and as you probably know, I follow
you.
what you do. It's a really wonderful talk, and I think I'm kind of thinking. Maybe let me ask my question. So first of all, the acidic SMAs. So does pH in mitochondria allow the activity of this enzyme, or do you think the acidity changes because of the disease, for example, and then it's more active? How do you imagine that? That's my first question.
A very good question. I didn't touch upon that. It wouldn't be on the inner membrane. It would be on the outer membrane or in the intermembrane space where pH is actually quite high. So inner membrane doesn't have acid single myelinase, and that way it's expressed and where ceramide could play a role is most likely in the formation of, in the release of cytochrome C and formation of the pores on the outer membrane of the mitochondria. So yeah, good question. Thank you.
So okay, so then maybe the two next questions are kind of together. So first of all, did you try to add and let me say this VLC 3-omega hydroxyceramide to cholesterol and see whether the cholesterol will crystallize? That's one. And then second, did you try any type of lipid supplementation for cholesterol crystals? Maybe even lanosterol. I don't know. I'm just thinking about how to dissolve them using maybe exactly the VLC 3-omega. Maybe this is connected.
I did not think about that connection about the omega hydroxyceramides and cholesterol. Maybe. No, we didn't do that experiment, but we can try. We can definitely look at that. And with the other lipids dissolving cholesterol, I think you're right on point there. I'm pretty sure that we could probably change lipid environment and help to dissolve cholesterols. We haven't done this experiment exactly that way, but I know that when we use the diets rich in fish oil, or diets that are rich overall in N3, we do have improvements of the retina, and clinical trials also support that. And we don't know exactly, there are a lot of mechanisms, but we don't know exactly what's happening. So that could be one part in there, because again, as I was answering Jim's question, that I think any lipid that is in proximity to cholesterol will probably help dissolve it. You just need to have that interaction. You just need to have that change in the environment that helps dissolve them.
Chris Kenny.
Hi, Julia. That was really a terrific. I learned a lot about lipids.
Thank you.
But I want to bring it back to mitochondria a little bit.
Of course.
Of course. When you were talking about the mitochondrial ceramides, And you indicated they're changed in diabetes. Have you looked at the mitochondrial ceramides in other diseases? Or can you give us a little more information about mitochondrial ceramides?
I have not, but there is a group in South Carolina, that Novgorodov and Gruz, they're looking at ceramides in inflammatory diseases and in cardiovascular health. Atherosclerosis, they're looking at cardiac muscle and endothelial cells, mainly around the cardiac aortic endothelial cells around there. So that's actually, I kind of almost copied their work because they did the studies first. And they also have another group there that's looking at the ischemic brain and stroke injury, and they're showing that there are changes in acid sphingomyelinase and ceramide in the mitochondria. So I haven't, but yes, there's some literature out there on that I looked at when we started, and I talked to these investigators, and they did show changes in. Actually, it's not just acid sphingomyelinase. There's a number of single lipid enzymes in the mitochondria, in the outer surface of the mitochondria, in the outer membrane of the mitochondria, outer membrane of the mitochondria, and that's where when you have pH change, you could have it active, and it could contribute to cytochrome C release and apoptotic changes.
Okay, thank you very much.
You know, I want to compliment you, Julia. It's a fabulous presentation.
Oh, thank you.
Certainly it's an area that I haven't followed, and I'm amazed at the progress. Do you have any idea what the mechanism of the fenofibrate positive response is that you see in the retina?
Well, that's the fenofibrate is the one that works in clinical trials, and we don't know why. We know that it's not because of triglycerides. We know that there's no connection to triglyceride and improvement of retinopathy. So what we see in our cells is that it dissolves cholesterol crystals. I have no idea why it's doing that. It was a big surprise to us. We actually tried it just because we're like, well, fenofibrate works in the retina. Let's see what it does. And it worked better than statins to dissolve crystals. How does it do it? I don't know yet. Again, probably just by being lipid soluble drug and attracting, like by hydrophobic attraction and the hydrogen forces, just dissolving, taking cholesterol off the surface of the crystal. But that's as much as I can tell you. And again, we are trying to work with chemists to see what can we do to understand what is required for the dissolution, what dissolves crystals. But it's very fast. We see it, like when we apply it, you can actually watch it with your eyes, how the crystals start kind of disappearing.
So do we know if there's any fenofibrate in the retina itself, or is it a secondary effect, do you think?
Oh, no, it's in the retina. It's again, we do not see any effect on the blood levels. Like when we look at
Oh, I'm sorry, we lost you. That was a fabulous presentation, Julia.
it's on like typical triglyceride. Oh, you lost me?
Now we have you back.
You were frozen for a second.
Oh, I'm sorry. Are you okay now? Do you
Yes.
see me? Okay. Yeah.
All right,
So
let's move to the final question, Dominique, and then I will just make a final comments, and we move on with the experiments of the day.
Dr. Busik, great talk. I have a couple of questions, if time allows. I was wondering, did you try to combine two methods, the one that you mentioned, anti-ceramide antibody fragment combined with acetic sphingomyelinase inhibition, to see if there will be synergistic effects of the lowering of ceramide levels?
We did. And whenever you inhibit acid sphingomyelinase, you are going to damage the neuronal tissue. And inhibition of sphingomyelinase by itself in the retina is not something I would ever advise doing. We had detrimental effects from that. And that's published in the literature that when sphingomyelinase is inhibited, you don't have normal lysosomal metabolism, and that's crucial for the neuronal tissue, for the support of neuronal tissue. Like as I mentioned, that the acid sphingomyelinase knockout animals were created as Newman-Pig disease model, and they do have severe neuronal damage. We did not do the inhibition of acid sphingomyelinase and in the bone marrow and the fragment in the retinal studies, but we did try it with the progenitor cells, which I just didn't have the time for that story today. So if we inhibit sphingomyelinase in progenitor cells, we actually don't have formation of this ceramide-rich platforms. So we applied the antibody after that, but we didn't have any additional effect, because if we don't have the formation of the platforms, we really don't have anything to inhibit with the antibody. So yeah, a good question, but I think that's...
Yes, but actually, so I'm not even talking about the complete inhibition by genetic knockout, let's say, but rather a pharmacological inhibition, which will bring you, give you some percentage of decrease in the hydrolysis of sphingomyelin by the sphingomyelinase, let's say like 20%, maybe 30% if you're lucky. And then, so you still have the activity of the enzyme, but it's just lower. Did you guys look into that, or rather just genetic knockout plus the anti-ceramide antibody?
We didn't, because I don't know of any acid sphingomyelinase specific inhibitors. You can use disipramine or amitriptyline
for
to inhibit it, but it's it will inhibit the acid as well, but it does it by destroying the lysosomes. It's not a specific inhibitor. And I don't think that they exist, at least like if you know, let me know, but I don't know of any specific acid sphingomyelinase inhibitors or small molecules inhibitors that can be used that way. So yeah, it would be great experiment to try, but it just I tried
desipramine, and it gave pretty good effects for lowering, but in combination with other drug, which is like for ceramide de novo synthesis. Yep. And actually about de novo synthesis, I have a quick question. Do you think, to your knowledge, which pathway is more important for genetic the ceramide pool in the cell, in the retina? Is it maybe acetic sphingomyelinase and sphingomyelin hydrolysis, or is it also de novo generation of ceramides, or maybe salvage pathway? Which one contributes to the more to the ceramide pool?
It depends on the length of the ceramide you're talking about. For 16-0 and 18-0, it's definitely sphingomyelinases. The salvage does not contribute much to it, and most of the ceramide that generated by de novo very quickly put away, because the activity of the sphingomyelin synthase is very high in the retina. So that pathway is really directed towards production of sphingomyelin, and then that's converted back to ceramide at the surface of on the membranes when ceramide is needed. That's we showed, and several other labs did demonstrate that. However, if you're talking about very long chain ceramides, then it's a ceramide synthase. Then that pathway, they are not acted upon by sphingomyelinases, and you don't make very long chain sphingomyelins. I haven't seen.
So you say mostly
maybe we stop on that, and if you have more questions,
sure,
that would be a way to contact.
Thank you very much.
Okay.
I have a final thoughts, but then it really came to my question, more specific. You decided to use alpha cyclodextrin. Why alpha, not the gamma or beta?
Very good question. Gamma and beta were not used in clinical trials because they're very toxic. They're actually better at dissolving cholesterol, but they're almost too good at dissolving cholesterol. they're removing it from the actually
Yeah.
from.
But
from
that's the
Mhm.
problem with alpha, that it is really not very good in extracting of cholesterol compared to the other cyclodextrin. And they will be loaded with other lipids rather than with cholesterol. This will, you know, there are kinetic study done in Langmuir where they study very specifically of extraction of lipids from one cells and transport to another. And the specific, I'm just surprised because it would be my last choice to use for cholesterol alpha cyclodextrin rather than two others.
Mm-hmm. It was one that we could use long term in animals without any issues, and it did dissolve crystals. We did see that it's dissolving crystals, and I wouldn't recommend using that. This wouldn't be the clinical trial choice for me. It was just something that we could do in animals, and we wanted to see how it would work. I think that fenofibrate would probably be my choice after seeing what it does and how well it works and dissolves that. But yes, it's just with beta, we couldn't keep the animals alive for that long. I don't know if what's your experience, but it just, they're losing hearing, they're losing vision, and they also have very severe diarrhea from beta.
Yeah, I don't think this is a good approach. You know, the lipids are extremely tightly controlled in all organs, and to changing even few percent of lipid content between you know, different experimental condition is quite detrimental for many reasons. And you know, to play with that to extract some sort of lipids, I found this unjustified.
Well taken. No, and that's not something, again, that's not something, I hope I didn't sound like I would recommend it to go into clinic. This was more animal use approach. Can we do it?
It has been also reported, as you know, to extract A to E, which I do not know even how that is possible.
Yep.
It's just intracellular deposits, which are completely insoluble, how they can be taken up by alpha cyclodextrin is something else. All right, we had a great conversation, a lot of questions. I'm sure we can contact Julia for additional questions if we have, and I will see you a week from now.
Okay, thank you. Thank you very much.